Okay, so we're in at one of our study sites in the Cypress Hills in just near Elkwater, Alberta, and this is one of a series of sites where we study the phenomenon of zombieism in ants and the phenomenon of host manipulation. And this is one of our main study sites, and it's really convenient because it's close to where we camp. And so this nest, this is a large nest in a stump, probably of a spruce tree. This nest contains a large colony of Formica acerba. There's certainly in the tens of thousands of ants in this colony. Now, the prevalence of infection in this anthill is probably about 5%. So 5% of the ants are infected. So infection levels are pretty consistent, and they're typically low at around 4 or 5%. But if you look at the vegetation surrounding maybe a meter or two of each nest, including this one, there will be ants attached to plants. You can see that this is a really complex system. There's lots and lots of plants for ants to choose. And one of the things that we're interested in is what plants ants actually select to attach to. And here's some examples. And so in this area, we have snowberry, we have horsetail, there's lots of grass, there's some Saskatoon berry, but there's also lots of dandelion. And it turns out at this time of the year, dandelion is one of the preferred plants. And here's a dandelion. This would be a plant, and here's some flowers that are senescing. So we're about just over a meter away from the nest entrance. And here you can see some attached ants. So here's a dandelion leaf. So there's an attached Formica acerba, so its mandibles are attached to the plant. This is really common as well. We tend to see infected ants aggregated on plants, and so they tend to select a plant and then bring their sisters to it. And so there's an example of, that's 1, 2, 3, 4, 5 attached ants on the underside of that leaf. Really, really consistent. But this is a much more common pattern that we tend to see, especially early in the morning, is we can see up to maybe 20 or 30 on one plant. The first host, don't forget, of this liver fluke are snails. This is a snail that's used in this area. This is Oreohelix subrutus, and they are absolutely incredibly common, particularly in a habitat like this. So you can see these are dead shells here, but these are all Oreohelix subrutus, and they're right underneath a plant that the ants are using for attachment. And so where we tend to see high infections are areas where we tend to see high snail densities. And then one of the other players, of course, is the final host, and that's in this area it's almost certainly white-tailed deer. So cattle aren't allowed in this area. They're fenced out, but cattle are another host, and elk are another host as well. And one of the most intriguing things, at least to us, is that the attached ants are on the one plant in this particular region that are obviously being grazed by, in this case, white-tailed deer. So that's classic evidence of grazing. And you can see this whole plant at one time, maybe a week ago, has been grazed. Of course, it begs the question, how many infected ants did that animal ingest? We know that every ant, every one of these attached ants, if we took them into the lab, took that ant and took it in the lab, it would have about 40 parasites in the abdomen, and it would have one parasite in the brain. And that parasite is in the brain in the areas associated with the action of the mandibles. And of course, that's the other thing that we do in the lab back in Lethbridge, is to try to understand how this whole manipulation works. And so I think what's really interesting here is the evidence that the ants are attached to the one plant of many around where we also see evidence for grazing. And the question, of course, is how that preference that the ants have, the infected ants, how does that manifest itself, is something that we don't know, but it seems really, really obvious.